네, 안녕하세요. Thank you everybody for coming out here today. It's a great pleasure to speak to you about our technology from Akoya Biosciences today. I lived in Korea for several years, so this really is exciting to me. So enough about me. I'm here to talk to you about Akoya's webinar, obviously, and we're going to speak today about the Codecs technology. And the Codecs technology is what we call a pragmatic solution for ultra-high-plex single-cell spatial analysis. That is the topic of what we will speak about today. So this is the Codex System. The Codex System was launched in 2019, and at the time it was already awarded one of the top 10 innovations by the Scientist magazine. It is the first and only benchtop solution for ultra-high-plex immunofluorescence with single-cell spatial resolution. So let's look at it a little bit closer. This blue box here, this is the Codex machine. It's essentially a microfluidic handling device. And this device integrates seamlessly with optical imaging systems, such as a Keyence microscope. You can see this here. This gray box is a Keyence automatic microscope, a BZX imaging series. And the Codex system is connected to this imager via this series of tubes that connect to the imaging stage. You can see the same thing here with a top view. So the technology is automated, and that's really important. So the experiment is very easy. Once you start it, most of it is automated. It's easy to use, and it's widely deployable. It's very small. It's low capital expenditure. It means it's not very expensive. So it is very easy to get into your lab, and it can be used for very many different experiments. So let's take a look at how the Codex experiment works. So when a researcher does this experiment, the first step is to develop an antibody panel. And with Codex, you can stain a single tissue with 40 or more antibodies. So you can see here is an antibody, this black thing, and then each antibody will be labeled with an oligo, a DNA barcode. When you do this experiment, you take your antibody panel, and in one step, you label your tissue with all of the antibodies at once. This is the only manual step you have to do. So you take all of the antibodies in solution, put them on your tissue, incubate it in the staining solution. I should note here that this is done at the moment on 22 by 22 millimeter cover slips. That is our recommendation. After the single step staining has been done, the machine, or basically the tissue, gets moved into the machine. So now the primary antibodies are on the tissue, and each primary antibody has a barcode on it. This is how you obtain your antibodies, or this is how you prepare your antibodies prior to the experiment. I'll speak about this in a second. Once it's in the machine will take reporters, 3 at a time. Red, green, blue. This is standard epifluorescence imaging. Each reporter will have an oligo barcode that matches specifically to a certain type of antibody. So antibody 1 will have barcode number 1, and reporter will only recognize that. Antibody number 2 will have barcode number 2, and the green reporter only recognizes that. Now, because it is standard epifluorescence imaging, you take an epifluorescence image with it, and then afterwards, the reporters from the first round are washed away. So you applied 3 reporters, you took an image, and you removed them. And then very simply, the system puts on 3 new reporters with 3 new oligo barcodes, and then takes a new picture. So essentially, this is an iterative cycling imaging where 3 fluorescence reporters are imaged at a time. And because they're recognizing different antibodies, every single time they're recognizing a different biology. That is how the Codex system works. Once again, you can do this with 40 antibodies at once in a single tissue, and what I've just explained to you happens inside of a machine. So you can go home at night, this machine will do this for you, and when you come home when you come back to the lab in the morning, you will have your data for 40 or more antibodies. This is an experiment, or this is just one example of an experiment in which a collaborator of ours stained human skin. So that's what human skin looks like when you label it with 7 different antibodies. You can see that antibodies are indicated here on the bottom. Most of these are for immune cell markers and keratinocytes. The reason I want to show you this image is to explain to you a little bit more about how the reagents work. So one of the things that's really easy and really nice about this system is that all of our reagents are available as kits, labeled, aliquoted, branded. You buy the reagents. You don't have to make your own reagents. We have Codex Antibodies. So if you're an oncologist or you're an immunologist, you're interested in immune system or cancer biomarkers, we have a lot of antibodies that are already inventoried for you and barcoded for you that you can buy off the shelf. We have reporters. So the reporter, of course, recognizes the antibody, but then also we have only barcodes. So a lot of people who use Codex is still a very new technology. They want to start a new application. So for example, you want to be, I don't know, study new field of neuroscience. You can take your own antibodies and you can combine with the barcode to make a Codex antibody. So you can use whatever antibody you want and you can do this experiment if you buy these barcodes. So all of the reagents are available from Akoya. We have both inventoried and custom ones. And this is what this table here shows you. So this image in the background, once again, this was an experiment that we did on skin. And everything that is red, these are antibodies that are inventoried. You can see a lot of immune cell markers. What is black here are custom antibodies. Researcher decided they wanted to use CD63. We didn't have it inventoried, but they took our barcode, combined it with this antibody, and then it became compatible for the Codex experiment. So that is a relatively quick overview of how the technology works, just so you get the idea. Essentially, it's ultra-high-plex immunofluorescence imaging. You can do 40 or more antibodies at a time. These are the facts that you would like to remember. So I'm an application scientist. I joined Codex with years of experience in imaging, and my job is to explore Codex and to see how you can use Codex in different types of experiments. So what I'm going to talk to you about today are actual different experiments and different types of data and different results. So you get a better idea, or so I can convey to you a better idea of what you can actually do with this technology. And it's really exciting. So the applications arena of Codex, you can work if you're interested in cancer discovery, immune oncology, immunology, anything. We found that the Codex system performs very well. So I'm going to explain 4 different types of experiments to you. 2 of them are related to cancer. One is a methodological one about how you can image more with less time. And one is a non-spatial biology application for immunology. And I'll go through these step by step. So the first thing I would like to tell you about or explore together with you is an experiment on which we did single-cell discovery in breast cancer. So this experiment was done in human formalin-fixed paraffin-embedded breast cancer tissue. So if you look at cancer papers today, a lot of the cancer researchers are very excited and very good at using single-cell biology techniques. So for example, single-cell RNA sequencing is extremely important technique nowadays because it allows you to take a tissue like a breast cancer and break it down into the individual cell populations. That's what you see here. This is a t-SNE plot. Every one of these little dots on this plot is a single cell type. The coloring shows you what cell type it is. The actual dot is the individual cell. So you can see these blue cells. These are so-called basal cells. There are not so many of them, but they cluster together. You see these red cells. These cells express E2F1. There's quite a few of them. This is what single-cell biology can do for you. Histology traditionally was not able to give you that much detail on a tissue. Generally, people look at 1 or 2 or 3 biomarkers at a time. But what I would like to show you today is that with Codex, this here is a Codex image in which we stain the tissue with 36 biomarkers, is indeed able to generate data that are deep and complex enough to generate these kinds of clustering analyses. This is really important. So you do single-cell sequencing, you're used to generating deep data sets. You're used to generating big data sets. With histology, you can now also do this with Codex. You can generate t-SNE plots, and you can, at the very same time, keep the spatial information of your tissue intact. You know exactly where every cell is. And that's the nice thing about Codex. So this t-SNE plot will be the topic of the first discussion that was actually generated with our Codex system and our software. So here you can see some data from our first experiment. This is human breast cancer, 36 antibodies we put on here. And I'm just going to go through here now, and I'm going to show you different images of the same tissue each time. Down on the bottom, you can see the actual biomarkers. So there are 7 biomarkers here. That's already more biomarkers than you would do in a conventional immunohistochemistry experiment. On the left-hand side, you will recognize that this is a relatively big tissue, 3.5 millimeters by 1.9 millimeters. Now, if we image the same tissue in multiple Codex cycles, you recognize each time a completely different type of biology. Like, you have so many markers at your disposal that you can really study not only the immune system, but you can also study the epithelial composition of this breast cancer. You can ask many questions in a single experiment in a very large and intact tissue. And that's exactly what Codex does. So the question becomes, how do you analyze this stuff? So we do everything based on cell segmentation. So we have a software that's called MAV, or Multiplex Analysis Viewer. This software will automatically segment for you all of your data, where every single cell is identified, as listed here. You can see cell number 1, 2, 3, 4, 5, all the way to 63,056. That's how many cells we identified in the tissue that I just showed you. Now, for every cell, you're going to have an antibody labeling pattern. So cell number 1 labels with antibody 1 at this intensity, antibody 2 at that intensity, and so on and so forth until you get to antibody 36. Anybody here in this audience right now who does sequencing will recognize that these data are not so different from what you would get in, say, a single-cell sequencing experiment. The big difference here is that we have spatial coordinates with single-cell resolution. We know exactly where every cell is. So what do we do, and what can you do with the Codex software? We have built-in clustering algorithms that allow you to take these complicated data sets. Again, you're talking about 63,000 cells at 36 different antibodies and spatial coordinates. That's complicated. If you're not a bioinformatician, it's overwhelming. We built in k-means clustering, so basically unsupervised clustering algorithms that allow you to organize your data and generate cell phenotype maps that can tell you what individual cell types are. So the computer obviously doesn't know what the phenotypes are, what their meanings are. You have to assign that, but the organization and the clustering is built into our software, and it works very nicely. So let me give you an example now. What you can see here on the bottom left is a t-SNE plot. In this t-SNE plot, I'm representing the expression of keratin 14. That's a biomarker for an epithelium. In red, you can see cells that are strongly expressing keratin 14. In blue, you can see cells that are only weakly expressing keratin 14. If you overlay this representation onto the tissue, essentially this tissue that you see in the back in pseudo color is exactly what you see in your t-SNE plot. You can see these red cells over here. These are represented in bright red throughout different types of epithelia in this breast tissue. This is what's called a tubular epithelium, or a collecting duct. This is where the milk in the breast is collected. The other ones are called lobules. They do not express keratin 14 so much. But what you recognize is that the expression of these different biomarkers is reflected on your tissue and on your actually clustered data. This is a very nice side-by-side overview of what you achieve when you do this experiment. Zoom out here. So let me jump over to the next slide. And this, of course, is then what a normal histological image looks like. So if we move on, you can zoom in here, and what you see here is one of these collecting ducts. And what we did in this experiment, just to confirm that our biomarker intensities and everything is represented faithfully, we measured the intensity of keratin 14. And you can see that in certain areas of epithelia, called the basal and myoepithelial layers, you get very strong expression of keratin 14. Whereas other types, luminal epithelia, this will be this up here, it's very weak. Now, this is just something we measured, but upon further investigation and upon reviewing the literature, we actually found that other laboratories that are completely independent of us, we didn't know about this result before, they find the exact same result as us. So what you can see here is a collecting duct. Again, this is a result from a different lab. On the top, this is a result that we generated internally, but look at the similarity between these results. Here's a collecting duct, it's bright red. Here are lobules, and here's our lobule. Our representative results, or our results, correlate extremely well with other accounts of how the breast tissue is organized. This shows many things. One of them is obviously that keratin 14 is expressed differently among different epithelia, but very importantly is that Codex recapitulates staining pattern very precisely in formalin fixed human tissues. And it can capture a range of biomarker expression intensities. So this is really important because it shows that the system, by employing a somewhat unconventional method for immunohistochemistry, can actually do this reliably. But it's also very important when you consider that our clustering and our analysis algorithm depends very much on fluorescence intensity. So what you can see here is a breakdown of all 36 markers in the experiment that I had just shown you. Each one of these is a t-SNE plot, and each t-SNE plot shows you the expression of a different biomarker. So if I toggle on here, you can see on the left-hand side, I'm showing the biomarkers keratin 17, 5, and 14. And go look over here in this middle column, you can see keratin 19, 8, and 8. These are different t-SNE plots. These are different representation of biomarkers. You can see a small population that's brightly labeled with keratin 17. You can see a pretty large population that's brightly labeled with keratin 19. So the next thing I would like to talk to you about is clustering and identifying cell types. What you can see here is a clustered cell type identification that we generated based on these biomarker expressions that I've shown you. And there are 3 cell types represented here just for the argument. First, you have a luminal cell type. There are 32,000 luminal cells in this sample. So you can see every one of these cyan dots all over here, those are luminal cells. Then you have 546 basal cells in dark blue. You can see there's some, but not quite that many. But then you have this tiny little proportion of cells of only 44 cells. That's 0.07% of the entire cell population that is here and here and here, and in a few spots, essentially. Now, I did this analysis. I'm not a breast cancer expert, so I didn't pay attention to these cells. These cells just presented themselves to us. So to me, the first question was, is this an artifact? 44 cells, nobody cares about 44 cells. It's such a small proportion of what's going on. Why is this showing up? So this is the beauty now of Codecs. This is really exciting to me as someone who used to do a lot of experiment with 3 markers. If you look here, we have a histological image. Here's our t-SNE plot, and here's a heat map that shows you the expression of 36 biomarkers for each one of these 3 cell types. So I went through this heat map, and I asked myself, well, are these cells unique? And if you look at this expression pattern, for example, now I added the marker TP63, you can see these yellow cell nuclei, essentially. They're expressed very nicely in what's called the basal epithelium. But there are still cell types left here that are not expressing this TP63. So you can toggle through this heat map of all these different biomarkers, and you will realize, for example, that beta-catenin, you can see here in blue now, is expressed in your luminal cell types. So if we go to beta-catenin, you can see this red square showing that the expression of this biomarker is restricted almost to this cell type. You toggle on, you'll recognize the same thing for TFAM. This is a transcription factor or a nuclear binding protein. You can see that is expressing in these types of cells, but not so much in others. So based on the density of biomarkers and the anatomical feedback that you have, you can be almost certain that what you're seeing here is not an artifact. Again, we're talking about 0.07% of the whole cell population. It's tiny, it's minuscule. But once we found it, and once we did this analysis with Codecs, we went through the literature and we realized that someone had already published this result before. So these cells are real, they exist, and we discovered them essentially on accident. So this is the point that I would like to make here to you, is that Codecs allows you to do very precise and unbiased phenotyping in formalin fixed paraffin embedded tissues. This is discovery. This is whether or not you're doing hypothesis-driven research or if you're really just doing descriptive research. This is human tissue that we got from a biobank. We had no preconceived ideas. We just labeled it with 36 antibodies that we were interested in, and we let the data guide us. And what we ended up with is discovering an extremely tiny population of cells that you would not find, for example, if you had to restrict ROIs or something, or if you had few antibodies. But we found it. We confirmed that it exists, and we are not the only people, or we're not the only research group in the world who has discovered these before. Turns out that these cells may actually play a role in etiology of breast cancer generation. So these are inherently interesting things. Again, this is a discovery tool, and what we've just done, we've discovered a small group of cells. So that was the first experiment that I would like to talk to you about. And I hope I've shown you one aspect of how Codex works. Now we're going to switch gears a little bit. We're going to stick with cancer research, but what we're going to do is we're going to kind of focus on how you can use your data in a more driven research question, and that is how to do cell phenotyping or immune phenotyping in a lung carcinoma. So once again, we're looking at a human tissue. This is a 3.5 millimeters by 2.6 millimeters large piece of tissue. Again, this is a fraction of how big you can image. You can image in total about 16 by 16 millimeter if you want. So this is just a small section, but this is a huge piece of tissue. This is a lung cancer, and you can go through here. We labeled this with 38 antibodies. And once again, as you're toggling through this tissue, each time you see a different biology. Each time something new arises. But in this particular experiment, we're interested in infiltrating immune cells. So I'm just going to step back forward. Here you can see all of your epithelia in cyan. And if I shave away this layer here, again, this is the same tissue each time. I'm just playing around with the different antibodies that we labeled with it. You can see all of the infiltrating immune cells. And there are many of them, and they're very colorful. So the question becomes, how do you categorize this? So one strategy that we've built into our software, once again, depends on your gating of cells. So you take this entire big tissue that you have here, you segment it down, and what you find after you segment your tissue is that there are 99,000 cells this time around. It's a lot more cells than what we talked about just before. And each of the cells will be broken down based on a singular label. So in this case, I would like to show you the gating function. So if you've done flow cytometry before, you're familiar with this. But what we do here is we take the intensity of say, for example, a pan T cell marker like CD45RO, and we plot it against the intensity of DAPI. And you can see in this scatter plot, each dot is a cell. The higher up on the y-axis these dots are, the more intensely labeled they are with CD45. So if you can, in our software, draw a gate around them, and you pick out all the intensely labeled CD45 cells. There's 2,611 of them, and you can see them here. So very easily, you can extract out all of the T cells that have CD45RO. It's very similar to what you would do in a flow cytometry experiment. Now you can go on and you can subgate these cells. So you can make a daughter gate, and you can take out all of the cells that are CD45. So once again, now you're plotting CD45 versus CD4. You make a gate, you pick out your cells, and you will see here in this little zoomed image, in red, you have your cells that are only CD45 positive. In white, you have your cells that are double positive. Now, what about CD8 cells? Same idea. You plot your CD45 versus CD8. You draw a gate, you pull out your cells. For each time, you get spatial coordinates, you get an intensity. Here's a negative cell, here's a positive cell, and it's always the same group of cells. So you've just immune phenotyped T cells based on their expression of CD45, CD4, and CD8. This you can do in our software while keeping an eye on your anatomical image, while having your gate in front of you, and while having your data in this FCS format. So you have tab data, you have morphological data, and you have these types of interactive viewers to work with. This is very efficient, it's very quick, and it translates back onto your clustered data. So everything that we've just identified via manual gating, we're plotting back onto our t-SNE plot now, and you can see that these two types of T cells form very distinct populations. So since we're at it, let's just look at the vasculature. Once again, we used a gating function for this, and we found that there are 2,899 cells that are expressing CD34 and CD31. These are your vasculature cells. Here you can see in green some blood vessels, And then let's keep going and let's add the keratinocytes. These are epithelial and cancerous cells in your lung, and they make up a huge proportion of your population. You can see this on this Tisney plot. There's 56,600 of them that are expressing this combination of different keratinocytes. So I've spoken about 1, 2, 3, 4, 5, 9 different markers. There are 38 different markers on this experiment, but this is already a lot of information. So what a lot of people want to do with this information, what I would like to show you quickly, is that you can very easily do spatial analysis. So in the case of immune phenotyping and immune cells in cancer tissues, there's often a question, how do certain types of cells relate to each other? So we have built in cross-correlation and Voronoi diagrams that allow you to break down the spatial organization of these cells. And more recently, we also implemented these Circus plots. So these plots show you the relationship between individual cell types. So you can see the cell types on the outside. This band here shows you the expression of a biomarker for each cell type. So let's focus, for example, on CD4 cells up here on the top right. You can see CD4 is white hot. It's like a fire. That's the strongest marker. So that is your CD4 cell. And then each one of these lines in the middle will tell you how that CD4 cell relates to another cell. So the thinnest band, the skinny black one, that shows you the weakest spatial correlation, defined by how close they are with the micrometer range that you enter. So you can see these CD4 cells, they're not really associating with your vasculature. What they are doing is they're associating with other T cells. Overall, this is a very handy tool to show you whether or not T cells are localized close to the stroma, close to the vasculature, or close to your tumor compartment. It takes very little time to generate this. It's built in. You do not need to have any coding knowledge to generate these kinds of data. Everything is built in, no code required. Essentially, you just follow a simple step of buttons. Excuse me for a second. So now that we went through this, essentially what I would like to summarize this with is that you start off with a really big tissue. You have 38 antibodies, you have 99,000 cells. All of these analyses that I've made here were generated in our software. Our software is free. It's a plugin for Fiji, so you don't actually have to buy that even. You can download that today if you like. And it's really, I think it has democratized the analysis of these complicated data sets because you have really zero coding knowledge required. I'm not a bioinformatician myself. I'm a histologist, but I've generated all of these data using the software. So if you don't have bioinformatician in your lab or you're not a bioinformatician, this should not worry you because it is very possible to do these analyses right now. Download our software, try it out. There are softwares and data sets available. So that's what I wanted to show you in the second section. Now we're going to talk about something a little bit more practical, more about the methodology of how this experiment is done. And that is that a lot of what people would like to do is they would look like a big tissue. They would like to image, 20 by 20 millimeter cancer tissue. But very often that's impractical. And what people would like to do is to image tissue multi-arrays. So this is an image here of an experiment where we work together with a lab to image 48 tissue cores in a single experiment. So each one of these circles is a 1.2 millimeter diameter core of a lung tissue. Some of them are cancerous tissues, some of them are normal tissues. They were put onto a single cover slip and imaged with Codex experiment with 38 antibodies. So every single one of these is on the same cover slip. It's running during the same experiment with 38 antibodies, 48 repeats. That really accelerates the throughput of your research. So here you have an example of 6 of these cores. I'm just showing you a fraction of them, core number 1, 28, 30, and so on and so forth. You can see your keratinocytes on your epithelial and cancer tissues. You have your infiltrating immune cells. You can see this very nicely. That's just a high resolution image of them. So you can see that the image quality doesn't suffer because we're actually using the same objective, the same lens for any type of our experiment. Most of the data acquisition that our customers do is done with a 20x lens. So it gives you decent resolution to do cell phenotyping. And then you can, for each single core, essentially make a little passport. Just like we've shown you, you can count your cells. So if you gate each core, you can gate cores together if you like. You can count your cells. So here, for example, you can see the cell counts for CD3. Here you can see the cell counts for CD4 type cells. Here you can see the cells for CD8. Each core can be clustered individually. You can see the distributions of individual T cells, or whatever cell you want to look at will be cataloged separately for each core. Then, of course, you can compare cores. Look at the difference in the histograms on the top. I'm just toggling back and forth between two cores. These two cores are different. This is what we didn't expect to see, but here we saw it. So the idea is that with Codecs, we now established that you can use a lot of markers. So you can look at a lot of different things. And you can do this with a very big tissue. So if you want to do discovery, if you're a descriptive biologist, or you don't really have a clear hypothesis, this is probably your best shot. Like, you can really, up to 15 by 15 millimeters, up to 40 markers, you can ask a lot of questions about a tissue. It's an incredible tool for that. It's really cool. It takes a while to process this, but it's really informative. Now, the issue here is, it takes, say, for a 15 by 15 millimeter, it probably takes you a whole day, a whole afternoon to do that. So you would have to repeat it over the course of a week. A lot of people that we work with, they have a more defined idea. It's hypothesis-driven research. For example, drug response versus no drug, or pre-drug versus post-drug, or this drug versus that drug. You can do many more tissues onto your cover slip. In this particular experiment that I just talked to you about, our TMA cores were 1.2 millimeters big. So they were still pretty big tissues, but much smaller. But we put 48 of these TMA cores onto a single cover slips. So if you think about it, you're increasing your ability to detect morphotypes, you're decreasing your runtime, your sample number goes up, you can add internal controls, positive, negative, whatever you want. It really expands the horizon of your experiment. So in a sense, the imaging, the analysis, everything is not really that different from one another. The TMAs are generated not by us, so there are services for TMAs. You would have to make your TMAs, but once you put them onto the cover slip, it's the same as you would for a large tissue. So that's what I wanted to show you about that. So I have a last section here now, and this is an application that we like to show because it's not a spatial biology experiment. So if you look down here on the bottom of this slide, Akoya Biosciences is the spatial biology company. So we like to look at intact tissues. We like to look at relationships between cells. But what I would also like to show you that if you have a Codex machine, you don't just have to do that. You can be unconventional. You can try something different. So in this experiment, we asked the question whether or not it is possible to use the same Codex machine that you use for what I just shown you to look at cell suspensions. So what we did is we took human peripheral blood mononuclear cells, so essentially blood cells, immune cells in the blood, spun them down, and we stained them with 26 antibodies against immune markers. And here you can see these cells. So here, 5 markers, and on this entire cover slip, we had about 600,000 cells. And if you toggle through each time, you get a different picture once again. Of course, there are different types of immune cells in a PBMC sample. T cells, B cells, macrophages, dendritic cells, killer cells. There's all kinds of stuff in there. And so with Codecs, you can take your cells, you can immobilize them on a cover slip. The cover slip is polylysine coated. You fix it, and the cells actually stick on the cover slips. They don't wash away. We get extremely good retention. And then you can segment your cells, just like we did, just like I've shown you. Each cell is identified. Each cell has an antibody labeling profile. Now, of course, each cell has a spatial location. It's not always important in this type of experiment, but it's really important, once again, that you can take these cells, you can cluster them, and then you can phenotype them. So here you can see an experiment where we did deep phenotyping of PBMCs with 22 antibodies. So we find approximately 11 or 12 different types of cells in this sample. This is normal human blood, healthy human, no known disease. We have B cells, T cells, granulocytes, macrophages, killer cells, circulating epithelial cells. We have their numbers. We have their representation of different biomarkers. Here you can see 22 biomarkers across, plus DAPI, each cell number, and then you have the representation on a t-SNE plot. This is for one sample. And we do this, once again, by unbiased clustering, and we followed or we developed a workflow that was established previously for cytoph and flow cytometry. We kind of merged these two workflows together and adapted them for our Codecs experiment. Now, if you look at the comparison between two different samples, so the two different types of human blood, both normal, here you can see the proportion of cells in color is different cell type. 100 means proportion as a whole. You can see they're relatively consistent. So you have the same number of B cells, CD3 T cells are similar. There's a bit of a difference in your CD4 cells, but other than that, they look very similar. This is also shown on your t-SNE plot. So Codecs is not only good to take a tissue and analyze its neighborhoods, do immune profiling in tissues, or do discovery in breast cancer research. You can take cell suspension, dissociated cells if you like. Maybe you can even grow cells in a cover slip, and you can label them with up to 40 antibodies at a time. You segment your cells as you would, and then you analyze, you phenotype your populations as you had done before. And that, in conclusion, says that Codecs is an incredibly versatile tool. So you have essentially an optical imaging-based system in your lab. You can put tissues on it, you can put cell suspensions on there, and you can image them with whatever antibody panels you are interested in. So that being said, I am not going to talk about too many more applications because this tends to get overwhelming. I'm just going to give you a quick snapshot of where we're going and what's happening right now with people who are using Codecs. So we talked about today about cancer discovery in breast cancer. I showed you how you can analyze FFP breast cancer tissues and identify small proportions of cell types. We talked a little bit about immuno-oncology. So we were going through different types of T cells in a big chunk of lung tissue. Then I showed you that it's possible to increase your throughput or increase the efficiency of your analysis by using TMAs. And then finally, I just spoke about using Codecs to do deep immune phenotyping of cell suspensions, which is a non-spatial application. Those are the things you saw. The things I haven't talked to you about in depth, but are going on at an equally important or more important degree right now, we are actually seeing a surprising amount of dermatologists that are using Codecs. So dermatology is an interesting field for immunology. And you can see this here. This is a skin tissue. I think I showed that at the beginning of the presentation. But this is actually a skin rash. And everything that's yellow or gold here are immune cells that are infiltrating these upper layers of the epidermis. This is the outside, this is the inside. And these immune cells coming out of the vasculature, and there's something going on in the skin. And people know very little about dermatitis or skin rashes. So there's a good effort going on at trying to actually understand the different types of immune cells that are here, how they're coming about, where they are, and where they're going. And Codecs turns out to be a brilliant tool for this. Neuroscience is really starting to take off, so that excites me. I'm actually a neuroscientist by training. So we started to image human brain tissues, mouse brain tissues, and the results are fantastic. So any one of you who are neuroscientists, we're presenting a poster at the European Neuroscience meeting in Glasgow. It's a virtual meeting, so you can just go online. That's actually happening this weekend. And in this meeting, we will show an experiment where we use 22 antibodies for neurons and glia and vasculature to stain the human cortex. Results are really beautiful. And that's what you see here. That's human cortex, yellow are glia. In cyan, you see synapses, and in red, the vasculature. Then finally, and evidently, Codex is a good tool for immunology, but this is not human tissue, this is a mouse tissue. And I like to show this picture to emphasize that Codex is obviously not only good for human research, it's also good for R&D mouse research. So it works in man and mouse. We've tried that, and we get good results for these types of experiments. And then lastly, a bit of perspective that we like to convey to our audience is everybody knows what this is today. coronavirus is, especially in the United States. I admire Korea for being, very good at this. But in the United States, we have a big problem with coronavirus right now. But there's also a lot of biology that needs to be determined now. And a lot of people are interested in studying biology of coronavirus. So with Codecs, you have a lot of immune markers. And we are now developing, or we're now implementing markers for COVID spike proteins, ACE2 receptors. These things are starting to come in now. So imagine doing an experiment where you can see your virus, you can see where it binds to, and you can label it with 38 antibodies for immune markers. That is an extremely comprehensive and extremely exciting, but also today a very, very important advance in science, because we need to understand what is going on with this disease. And Codecs, you can study the lung, you can study PBMCs, you can study the brain, you can study the skin. All of these things are affected by this disease. So we think that our platform could be a very pragmatic solution to advancing research in this area. And with that, I'm going to now conclude my part. And I really want to thank you for watching from our side here in Menlo Park, where Akoya is. And of course, I will mention our partners in Korea, BMS. They will be there for you if you have any requests after the Q&A session. So thanks again for listening in. And thanks everyone who set up this webinar. We're super excited to be here. And we're really, really excited to have had the opportunity to speak to you live. So thank you very much.